Welcome to our video presentation on the groundbreaking research conducted by the University of Oxford, revealing fascinating insights into how antimicrobial resistance emerges in patients with bacterial infections. Published recently in the prestigious journal Nature Communications, this study's findings could potentially revolutionize our understanding of antimicrobial resistance and guide the development of more effective interventions to combat this global health concern. Traditionally, it was believed that bacterial infections resulted from a single genetic clone or strain of pathogenic bacteria, and that resistance to antibiotics developed due to new genetic mutations during infection. However, this new research challenges that conventional view. The study suggests that patients are often co-infected by multiple pathogen clones, and resistance arises from pre-existing resistant clones rather than new mutations. To investigate this phenomenon, the researchers used an innovative approach. They studied changes in genetic diversity and antibiotic resistance in a pathogenic bacteria species called Pseudomonas aeruginosa. The study involved samples collected from 35 ICU patients across 12 European hospitals. Close-up of Pseudomonas aeruginosa. Pseudomonas aeruginosa is a notorious opportunistic pathogen responsible for hospital-acquired infections, often affecting immunocompromised and critically ill patients, leading to more than 550,000 global deaths each year. Each patient was screened for Pseudomonas aeruginosa upon admission to the ICU. Samples were collected at regular intervals, and a combination of genomic analyses and antibiotic challenge tests were conducted to quantify bacterial diversity and antibiotic resistance. Surprisingly, the findings showed that around two-thirds of patients were infected by a single Pseudomonas strain, and resistance developed due to new resistance mutations. However, the remaining third of patients were co-infected by multiple strains. What's more intriguing is that resistance increased by about 20% more in patients with mixed strain infections when treated with antibiotics, compared to those with single strain infections. The rapid increase in resistance among mixed strain patients was driven by natural selection for pre-existing resistant strains that were already present at the start of antibiotic treatment. However, the study also suggests that while antimicrobial resistance might evolve more quickly in multi-strain infections, it may also be lost more rapidly under these conditions. When samples from both single and mixed strain patients were cultured without antibiotics, the antimicrobial resistant strains grew more slowly compared to non-resistant strains. This implies that resistance genes carry fitness trade-offs and are selected against when antibiotics are absent. The research indicates that interventions aiming to limit bacteria spread between patients, like improved sanitation and infection control, could be more effective against antimicrobial resistance compared to measures targeting new resistance mutations. Additionally, the study suggests that clinical tests should focus on capturing the diversity of pathogen strains and infections, enabling more accurate predictions of antibiotic treatment success, similar to how cancer cell population diversity predicts chemotherapy outcomes. Craig McLean, the lead researcher from the University of Oxford's Department of Biology, emphasized that the key finding of this study is how resistance rapidly evolves in patients colonized by diverse Pseudomonas aeruginosa populations. These insights could have a significant impact on our approach to combating antimicrobial resistance across various pathogens. This issue is particularly critical as the World Health Organization has declared antimicrobial resistance to be one of the top 10 global public health threats. It's associated with nearly 5 million deaths worldwide, especially due to the rapid spread of multi-resistant pathogenic bacteria that can't be treated with existing antimicrobial medicines. Professor Willem van Schaik, director of the Institute of Microbiology and Infection at the University of Birmingham, highlighted the study's implications for clinical diagnostics and infection prevention efforts. Researchers working in a laboratory, he stated that diagnostic procedures might need to consider more than one strain from a patient to capture genetic diversity and antibiotic resistance potential accurately. Professor Sharon Peacock from the University of Cambridge emphasized the importance of infection prevention and control measures in ICU settings, emphasizing that reducing the risk of acquiring pathogenic organisms during hospital stays remains a critical aspect of patient care. In conclusion, this study has brought to light a revolutionary understanding of antimicrobial resistance and its emergence in patients with bacterial infections. By shedding light on the complex interplay of multiple pathogen clones and pre-existing resistance, Researchers hope to pave the way for more effective strategies to combat this global health challenge. Thank you for joining us in exploring this fascinating research. For more information, please refer to the provided source link. If you found this video informative, don't forget to like, share, and subscribe for more engaging content. Stay informed and stay healthy.